Hello, everybody. Welcome to another episode of CellTalk, where we will be talking all about science. I'm your moderator, Dr. Chiku Cherian, and today we'll be having with us Nupur Biswas. Nupur Biswas is working as Senior Bioinformatics Scientist at Renix. She's leading the bioinformatics research team and is responsible for monitoring multiple projects. Some of her projects include designing personalized neoantigen vaccines for cancer patients, exploring the genes behind chordoma and health applications of digital healthcare wearables. Nupur holds a doctoral degree in physics. Her research was focused on studies of confined polymers. Prior to joining Renix, she has worked at the Indian Institute of Science, Raman Research Institute, Collabtree, and Indian Institute of Chemical Biology. Welcome, Dr. Nupur. Now I hand over the stage to you.
Thank you, Chiku, for this nice introduction. Okay, let me start sharing screen. Hi, everybody. I am Nupur from Renix Life Sciences. So today I am going to talk about one of research article. We will discuss about the result from one research paper. At Renix, one of our goal is to interpret, to translate the research outcomes to the end users. So this talk or this discussion is a one step towards that mission. So today I will discuss about the role of two genes, EGFR and ADD3, how these two genes are determining the subtypes of the glioblastoma. The original work was done by Lara Navarro and Teresa San Miguel, and there are other authors also, but these two persons are the first authors of the work. This work was published in the journal Cells in the year 2020. This journal has an impact factor of 6.6, and the title of the work was Identification of New Genetic Clusters in Glioblastoma Multiforme, EGFR Status and ADD3 Losses Influence Prognosis. And here is a snapshot of the paper. So we see there are multiple authors are involved. Lara and Teresa are the first authors, and Concha, Miguel, they are the lead authors. And this group is from the Spain, and they did the work. It is a combination of both experimental work and bioinformatics analysis. So it's a nice combination. The work is focused on the glioblastoma. So first of all, let's talk about what is glioblastoma. In one sentence, glioblastoma is a brain cancer. It starts from a type of tissue, glial tissues of the brain. Glial tissues are the supportive tissue of the brain, of brain's nervous system. So glioblastoma occurs in different parts of the brain, but mostly it occurs in the frontal lobe and brainstem part. And the cell from which most type of the glioblastoma occurs is the astrocytes. Here this picture shows how the astrocytes is linked with the nervous system. The neuron is here, and this is the blood stream. So astrocytes maintain the connection between the neuron and the bloodstream. Basically, they control the blood flow to the nervous cells. Now, when the cancer occurs, the astrocytes turns to the cancer cells, and these are the glioma. These blue cells are the glioma cells, the cancer cells, and they disrupt the blood supply to the nervous system. That's the way glioma affects its patients. Now, here is some statistics about glioma. The statistics based on the US population. Around 3% get affected by this disease among 100,000 patients. And this disease is quite uncommon in the children. The median is 64 years. That is quite the aged people suffer from glioblastoma. And this disease is quite prone to the males, 1.6 times higher occurrence in the males compared to the females. And also, this is more occur in case of the Caucasian populations to Africans compared to the African and Afro-Africans, and it has much lower occurrence in case of Asians and American Indians. Now, what is the issue with the glioblastoma, why it is a challenge. The main challenge is, of course, the location of the tumor. It is in the brain. So obviously, it's quite difficult to do the surgery. Although surgery is the major treatment for the glioblastoma, but that is difficult since it is localized in the brain. And there are other issues for that associated, that is the limited capacity of the brain to repair itself. And it has inherent resistance to the conventional therapy. And the other issues like migration of malignancies to the adjacent tissue, which we observe in other types of cancers also. And then the disrupted tumor blood supply, which actually inhibits effective drug delivery because most of the drugs are carried by the blood. And the resultant neurotoxicity of the treatments that makes the situation more complexity. But apart from these challenges, the major challenge is the genetic heterogeneity. Yeah. So we know the cancer is a genetic disease. It comes due to the genetic alterations. These alterations can be of many types. It can be mutation. It can be mutation. It can be amplification of genes. It can be loss of gene. But in case of the glioblastoma patients, there are lots of variations, genetic variations are observed. This genetic heterogeneity makes it very difficult to treat the patients because the patients differ a lot. To treat this genetic background landscape of the glioblastoma, I will discuss few genes which are very significant in the scenario of the glioblastoma. One is IDH. It is isocytosine dehydrogenase an enzyme which produces alpha glutarate, a molecule which promotes cell differentiation. So if IDH gets amplified, or that means it increases, it promotes cell differentiation, that is, it promotes in the cancer. But the mutant IDH actually can't make alpha Kg, and this can't promote cell differentiation, is actually helpful for the glioma patients. And mutant version actually acts as a good marker of prognosis for glioblastoma. But unfortunately, 95% of the patients are of IDH wild type. Now there is another gene which comes in the scenario of glioblastoma very often, that's EGFR, epidermal growth factor receptor. This gene often gets amplified in the wild type version, and also it has various mutant versions. The mutant versions also get amplified. In this paper, the researchers have focused on one particular mutant version, which is called EGFR variant 3, where exon 2 to 7 of the gene is deleted. This exon deletion corresponds to deletion of 267 amino acids from the extracellular domain of the receptor. And this mutant version has different signaling mechanism in the context of the disease. So with these things, the objective of the research work is to characterize the frequent alteration of EGFR by its amplification and the presence of the mutant version. And another objective is to associate somatic copy number alterations with EGFR amplifications and the presence of the mutant in case of IDH wild type patients. To explore these issues, this group studied, tissues from 128 patients. Since the samples were collected by surgery, these patients were not given any chemotherapy or radiotherapy when the sample was collected. And with these samples, they did one experiment, that is Mlpa, multiplex ligation dependent probe application, which gave them the copy number alteration information of some selected genes. Apart from this experimental approach, they did some bioinformatics approach like survival analysis, clustering analysis. They also access data from the TCGA database to validate that whether their results from their patients are similar to the patients from the patient data available in the TCGA. So before going to the result, let's very brief introduction about the Mlpa technique. It is actually a modified version of PCR. So we know that NGS technique, whole genome sequencing, exome sequences are helpful for investigate the genetic mutations in case of cancer. But these are quite expensive also. Mlpa is a variation of PCR where you can check multiple genes at a time. Right. So basically, there are multiple Mlpa probes are available. These probes are allowed to hybridize and then go for the PCR reaction using the same PCR primer pair. And then it gives the copy number alterations of the corresponding genes. So in this paper, the researcher used around 50 genes. And this is the result from the experiment for different genes. The x-axis shows the list of the genes and y-axis correspond to the amplifications, amount of amplifications of the genes. And the red, blue, yellow, these colored dots are correspond one sample. That is the patient data. So we see that these dots are distributed over a broad y-axis. That means there is lots of variation among the patients. That indicates the wide genetic heterogeneity among the glioblastoma patients. Still, among these genes, most altered genes are EGFR, CDKNA, TIMP3, like these are. To validate this and result, they also did analysis on the TCGA samples and got similar results similar results. Now they did some survival analysis to associate how these effects of gene alterations are on the survival of the patients. So they see the ADHwt gene. They studied ADHwt in particular because this gene also showed high alteration in the copy number. The patient with the copy number alteration in the ADHwt has lower survival time. And they also showed that EGFR wild-type patients have actually more survival period compared to the mutant patients. With this thing, they did how EGFR is related to the copy number alterations of may have some associated genes. Because we know in the cancer, often multiple genes play together, the multiple genes together determine the disease prognosis. Here also they noticed, the first graph shows that the copy number alteration related to EGFR amplification status. So this blue means the normal level of EGFR, orange means low amplified EGFR, and gray means high amplified EGFR. We see 4 genes because most important: MSHC, CDKN2A, MTAP, JAG1. Among them, let's concentrate on the CDKN2A one graph. So we see among the highly amplified EGFR patients, most copy number alterations occurrence was more. And then they also compare how copy number alterations related to the mutant version of EGFR. They are also see for most of the genes, at least the genes they have shown here, highlighted here, like CDKN2A, P10. Mutated EGFR is accompanied with higher copy number alterations of other genes. So mutated version is causing more alterations of the other genes. So there is an association between the genes. And then they went for the clustering analysis. The goal was this analysis is to see subtypes among the patients. If they find different types of patients having similar genetic modification, then these patients can be treated in the similar fashion. So clustering analysis was done with 73 samples, quite less number of samples, because some samples did not show up in the clustering analysis. That means they could not be classified. Right? So among the 73, they found 3 clusters, C1, C2, C3, then just named it. So number of patients within these clusters is almost in the same range. C1 has 27, C2 25, C3 21 patients. And this graph shows some green at red. Green means there is copy number alteration for the genes. Genes are listed here. I just highlighted EGFR here. And red means that there is alteration, and green means this is no alteration. And the row corresponds to the patients. So we see C1 samples at mostly normal level of EGFR. There is mostly green in this column. But the C2 and C3 are highly red. So they have most high EGFR amplifications. And other genes are also amplified highly in case of C2. C3 is a mixed case for the other genes. Now, let's discuss what this clusters is. Now, we divided the patients into 3 clusters, but there are further implications of these clusters. Cluster 2 showed highest frequency for alterations in CDKN2, MEN1, EGFR1, EGFR, TM3, this type of mentioned genes. These genes correspond to some pathways. They did pathway analysis to know what are the biological processes are relevant in this scenario. The pathway analysis told that the more association with the regular of cell surface addition process, cell matrix addition process, and cluster 3 showed the association with the regulation of cell cycle phase transitions. For the cluster 3, TP53, CSIX3, IL4, these genes becomes important. And the cluster 1 has the less altered cluster, not affected by EGFR. So, and almost similar number of patients are in the cluster 1. So that is an issue to be concerned, because we could not relate the genetic heterogeneity in case of the cluster 1. So the impact of the study remains that the wide genetic heterogeneity is challenged, and they tried to categorize this heterogeneity. EGFR wild type variant 3 with alteration in ADT3 has bad prognosis. So though this combination can be used as a prognosis marker, Mlpa technique can be considered as a diagnostic technique for the significant genes in case of which are already known in case of glioblastoma. And this technique can be applied for the other cancers also, as it is a cheap technique and easy to analysis is also easy. The cluster analysis has allowed characterization, but the issue which I mentioned earlier, the presence of group YDH wild type samples where EGFR alternation are not noticed. That's the cluster 1 group. So the association of EGFR and GBA wild type is still not linear. So more study is to be needed in this scenario. So that's all we can talk about this project, this paper. If you have more questions, you are very most welcome. And thank you everybody for your patience. And you can reach me later also at my email address is shared here, nupur@renix.org. Be free to email me whenever you wish. If you have any query or anything. Thank you, everybody. Thank you, Chico.
Thank you, Nupur, for that enlightening session on glioblastoma. So I hope we all could learn a lot about this disease. So there has been some questions that has been raised by the audience. First, we have Shweta, and her question is ADD3 loss a frequent alteration in glioblastoma?
Yeah. ADD3 loss is observed in case of the glioblastoma. And patients with ADD3, the alteration shows lower survival period. Yeah.
Okay. Her next query is SCNA a significant alteration as EGFR amplification or EGFR variant 3 in glioblastoma, or which is more significant?
Well, combination of EGFR wild type, combination of EGFR mutant variant 3 with copy number alteration, high copy number alteration is bad for the patient, I would say. That is of more concern. This combination has lower survival period.
Okay, thank you, Nupur. Next, we have Sauvik, who have raised 3 queries. The first one is, how many somatic number variations are found for the specific EGFR variant in the study?
Well, the number, how many somatic number variations, that is not shared. What is shared is this is copy number alteration. That is they have not measured probed the mutation in the EGFR. They have worked with only one mutant version, that is variant 3. So what they probed here, how much EGFR is amplified, not how many mutations are there. How much amplified means how much copy number it has gained or lost. It actually has gained, not lost. Okay, it's not the somatic variation mutation. It is copy number on alterations.
Okay, Divya, got it. No, sorry, Nupur got it.
Okay,
Thank you.
Okay, Sauvik's next question is, what are the advantages of multiplex PCR over NGS?
Okay, first of all, there is pros and cons of this method. Okay, so advantages, it probes PCR, but in NGS we check the whole genome or whole exon. So we look at the every genes. If all of the genes are not, of course, not important in case of cancer. In many cancers like glioblastoma, lung cancer, breast cancer, we know some genes which are observed in the which gets mutated or altered in the patients. So those particular set of genes you can probe in your patient. But if you want to study some particular list of this, then you can go for MLPA.
We should be knowing the at least the target genes.
You have to know the target genes. Yes, target genes.
Otherwise, NGS is the best option, right?
NGS is best option, but again, it is time analysis is robust, more robust.
Okay, so Sauvik's last query is multiplex PCR better over NGS for prognosis of cancer?
Yeah, if you to study prognosis, to check repeatedly which genes are getting. Suppose a patient is under medication, you want to check how a particular gene is getting modified, the effect is. Then you can go for the MLPA. Because PCR you can do one or two genes. Advantage of MLPA, you can do multiple genes at a time.
Yes. Okay, and it is cost effective also, right? MLPA method.
I hope so.
Yeah.
I hope so.
Yeah. Okay.
It should be.
Okay. Thank you for your answers.
Thank you, Sauvik. I hope Nupur could address your queries. Now we have Aditi, who would like to ask a question, like a generalized question. As a person being introduced to this kind of research for the very first time, can you explain what terms like EGFR, MLPA, NGS, and gene clusters actually mean in the simplest possible way?
Well, these terms which you mentioned are completely of different types of things. First thing is EGFR you mentioned. Okay. EGFR is a gene. This full form is epidermal growth factor receptor. It's a gene. Since it is a big name, we call it in a shortened name EGFR. It's a gene. Yeah, somebody.
No, it's me only. I'm understanding.
Yeah. And then you asked, I think, MLPA. MLPA is a technique. It's an experimental technique. Okay. There are different techniques to study patient tissue sample. PCR is one technique. MLPA is a modified version of PCR. NGS is the next generation sequencing. It actually probes the whole genome of the patient. That is another experimental technique. Okay. And any other term?
Gene clusters. Gene clusters.
Gene cluster. Okay. So what happens in case of the patient? Many times, not only one single gene gets mutated, multiple genes get mutated or changed at the same time. So these genes make a cluster. We call this group of genes are getting altered. That's why the term gene cluster is used.
Okay, thank you.
Welcome.
So thank you, Nupur, for addressing these questions. Now the stage is open for anyone who wants to ask a spontaneous question. So you can go ahead, you can unmute yourself and ask any spontaneous question you want, or you can write it in the chat box and I will read it out. Okay, so I think that was a very enlightening session by Dr. Nupur Biswas, and I hope we could have a good takeaway message from this presentation. So without further ado, I would like to end this session, and I hope everybody could benefit a lot from Dr. Nupur Biswas's lecture. So thank you so much and have a great night.
Thank you, Chico. Thank you, everybody.